Great. Once again, I'd like to welcome everyone to the Los Angeles Birders webinar. I would like to, excuse me one second, just my thing's getting weird. I'd like to introduce Tori Fay, our new chair of webinars, to introduce our speaker for tonight. Tori?
Great. Thanks, Ron. It's my pleasure to introduce today's speaker, Dr. Allison Shultz, Associate Curator of Ornithology at the Natural History Museum of Los Angeles County. A native Californian and lifelong enthusiast, Dr. Shultz is not only a leading evolutionary biologist, but also someone whose scientific journey has been profoundly shaped by curiosity, creativity, and a deep commitment to inclusivity in science. Growing up in Dana Point, her passion for the natural world began early. That spark grew during her undergraduate years when she worked at the Museum of Vertebrate Zoology at UC Berkeley and explored changes in bird populations while she was on campus. This experience opened her eyes to the power of natural history collections and the stories they hold about evolution, ecology, and environmental change. She then pursued a master's degree at San Diego State University and her Ph.D. In organismic and evolutionary biology from Harvard University, where she began exploring one of her primary research interests, how bird traits evolve over time. Today, Dr. Shultz leads a research program that integrates genomics, museum collections, and field data to uncover the evolutionary drivers behind feather coloration. As an artist and a scientist, she brings a unique perspective to this work, seeing vibrant feathers as beautiful and complex biological structures shaped by physics, ecology, and behavior. Her office walls, which you can see behind her, are filled with paintings and pieces of artwork brilliantly colored like tanagers and turquoise, a daily reminder of the questions that inspire her research. Beyond her work in the lab and museum, Dr. Shultz strongly advocates for diversity and inclusion in science. She actively works to broaden access to STEM fields, particularly for those historically excluded from them. She is also a champion of Los Angeles, the birdiest county in the US, and the need for urban gardens with native plants to provide oases for declining bird populations. Today, she'll take us deep into the fascinating world of feather color and tanagers, focusing on the mechanisms and modifications that produce their dazzling diversity. Please join me in welcoming Dr. Allison Shultz. Thank you.
Thank you so much for that wonderful introduction, Tori. So I am going to share my screen. Oh, yeah, now I'm going to share my screen. All right. Well, it is my really great pleasure to be here. I think this is my fourth LA Birders webinar that I've given.
I believe so.
Yeah, over the years. And so it's really fun to continue to talk about my research and talk about all of the exciting things that we have going on at NHM. And actually, this is the first time I've given this talk and talked about this particular project. So it was really fun to put together some new slides. So you're all seeing stuff that I haven't presented on before, which is really exciting. But before I get into the new stuff, I just want to do a little bit of a review of what I do and what have we found and kind of position us to be able to tell the story of what we're working on right now. So one of my main study organisms are tanagers. Tanagers are really exciting for studying evolution of plumage color. They come in every color of the rainbow, as you can see from this really fun wheel of tanagers that I've put together from our collection. They also come in all sorts of different browns and blacks, so they're really wonderful for understanding what birds are what colors when. So a lot of my inspiration comes from looking at tanagers and other birds in our collection. You know, what I'm going to tell you about actually stemmed from just curiosity about a pattern that we noticed when we were looking at some of the tanagers in our collection. And really kind of some of the key questions that drive me when I'm looking at all of these amazing specimens that we have is, why are birds the colors that they are, and how are birds the colors that they are? There are a lot of other questions that I think a lot about, such as how are birds changing over time? But for today's talk in particular, we're going to be focusing on the color side of things. So I'm going to do a little bit of a review of bird coloration for some of the talks that I've given here in the past. So, you know, bear with me if you've heard me talk before and remember all of this. But for those of you who perhaps don't think about bird feathers and colors all the time like I do, I think this will be helpful. So a lot of what we study are bird colors as we see them. They're the colors that we observe with our eyes and the patterns that we see. There might be different colors on different parts of the body. And so to study these, we take what are called reflectance spectra. So here is my friend, the paradise tanager. Paradise tanager will appear throughout the talk. This is a really brilliantly colored tanager. And I have a device that I point at the different color patches of the bird, and the readings go from those color patches into my computer and can tell me how much light is being reflected off at each wavelength. So remember, light is made up of different, its basic color is made up of different wavelengths of light and how our eyes perceive them. We'll talk a little bit more about that in a second. And so the higher the reflectance, the more light at that particular wavelength. So you can see this blue curve here, which corresponds to that blue belly of the tanager, has quite a lot of reflectance right around about 370 nanometers, kind of right about there. Interestingly, right as also about 550 nanometers. Usually we think of 400 as being kind of violet and blue. Kind of this range is about greens, this range is about red. So it's interesting that this blue actually has peaks both in the green range as well as the violet range that produce what we see as blue. And so how do we actually think about these reflectance spectra that we measure? Well, for a long time when people were talking about bird coloration, they would actually quantify birds as being colorful or not colorful. They might say that this bird is brightly colored or this bird is green or blue, but all that was according to the human eyes. Now, why is that relevant? Well, birds don't see the same way that humans do. So what I'm showing here are the sensitivities of our different cone receptors in eyes and comparing humans and birds. As humans, we have 3 different cones in our eyes, roughly red, green, blue, think RGB color. And so it's the relative stimulation of these different cones that send signals to our brains that are going to say that an object is a particular color. Now, birds have 4 types of cones in their eyes, so they actually have a whole nother dimension of vision that we don't have, and that 4th cone is actually in the ultraviolet part of the spectrum. So if we're studying bird color, we have to study bird color from the bird visual perspective. Now, so now I've got these reflectance spectra. I know how birds' eyes are sensitive to different wavelengths. Well, we can combine that into something that we call the avian tetrahedral color space. This is just a way to measure how colorful different birds are. Each of these different apexes represents one of the different cone values, and so the relative stimulation is where we plot each point from that reflectance spectra. So for example, this paradise tanager, we can take the volume of space that's occupied as like a way to measure how colorful it is without taking into account the biases of our human visual system, but study them according to how birds see them. So I've done quite a lot of work on this and using this way of measuring color. And that's kind of what got me started on my journey of studying color. But since then, I've been thinking about how are colors made from a mechanistic perspective. So I just talking about color as a whole. So we take these measurements and we can quantify plumage. But really, these patterns are made up of arrangements of feathers. Feathers are arranged particular ways on the body. And so we might also think about bird colors from a perspective of different regions of the body that make up that overall pattern. We can go even more detailed and look at individual feathers and think about what we call the macrostructure. So if this is a body feather of a bird, we can divide it up into different regions. We've got here the plumulaceous region, aka the downy part of the feather. This is the fluffy part that we think of as being associated with keeping birds warm. And we have the pennaceous part of the feather. This is typically the colorful part of the feather. That is what you're seeing when you're looking at a bird's color. Just for this, the rachis is what basically the trunk of a tree when we're thinking about the feather. And we have these little feather barbs that come off of the rachis. So remember that birds are divided up into the the downy part of the feather and the colorful part of the feather. That's going to be important in a little bit. We can get even more detailed. We might look at what we call the microstructures of the feather. So we might look at the shapes of these little branches of the feather. These little branches, as I said, they're called feather barbs. They're composed of a beta trunk with little twigs coming off of those. Those are called barbules. So we've got all sorts of different things that we can measure and think about how those are contributing to feather color. We can go even more detailed and look inside of these different parts of the feather at how the molecules are, oh, sorry, I was looking at the chat, just making sure there's nothing that I needed, how the molecules are arranged. And that's what we call the nanostructure. So that's actually very relevant to color because how light is scattered off of those different nanostructures produces blues, purples, ultraviolet colors, or iridescent colors. Here I'm showing two different examples of how those colors can be. One is the blue of bluebird. One key thing you notice, this is a close-up of the feather, and all of the colors, when we look really closely, are in the feather barbs. So you see the barbs here, whereas these are both iridescent colors, which are produced by a different mechanism. I'll get to the details in a minute. And if we look at the close-ups of the feather barbs, you see that the barbules are actually what are shining. So it's two different ways that light is scattering off to produce those colors. Now, what does that mean if we look at it closer? Well, if we look at the inside of a barb of a bluebird feather, you notice that there's this spongy layer that kind of looks honeycombed. Here's a close-up of that spongy layer. And it's actually the distance of those holes that light is going to be refracting off of. And so you can change the diameter of that distance, and that is going to be indicative of what wavelengths are scattered off of it. So that's how birds are tuning their structural color. Here's another depiction, which is showing how light will basically go into the feather. And in this case, blue light is scattered off and all of the other light is absorbed by this black melanin layer. Iridescent color, somewhat similar, except that in this case, again, these are in feather barbs and barbules instead of the barbs. Light is basically being reflected or refracted off of this melanin layer, which is basically just disordered scattering in something like a blackbird that looks like an oily sheen. But in something like a hummingbird, we got actually these very fine, organized layers of melanosomes that are going to be scattered at different angles. And so the key here is that light changes according to angle. I know that's a lot of details. The key things I want to take away from that are that nanostructures scatter light, and that's a way that color's produced. But then again, we can get even more in detail and look at the molecular composition. So birds can put chemicals inside of their feathers. Those chemicals we call pigments and actually absorb different colors of light. And so in this case, this particular pigment is absorbing everything but red. And so the red light is being scattered by this, this cortex, which if there was no pigment here, that would just be white because there's no particular wavelength that's being scattered off. It's all about what's being absorbed by the pigment. Some of the most common pigments are probably familiar. They're called melanin. They produce all of the blacks and browns and rufous color, the same as our hair and our skin. We have the same pigments in us. And some of the other most common pigments in birds are reds, oranges, and yellows, which are called carotenoids. And so these are some of the ways that birds basically use these different methods to make their colors. And they, one thing to keep in mind is for a long time, people talked about structural color versus pigmentary color, but all structural color relies on that melanin base layer in order to produce those colors because they have to absorb all wavelengths of light. And all pigmentary color is in some sort of feather structure that's going to have to scatter everything in order for it to work. This is one of my favorite examples. There's a really cool paper that came out in, I think it was 2005, where they looked at the feathers of a leucistic Steller's jay. And so they found that here's like a typical Steller's jay feather. This is that spongy layer. So this is that layer that's scattering the light. This is the melanin beneath it that's absorbing all of the light but blue. This is a leucistic version where they still have the same spongy layer, but they're missing the melanin. So think a bird that's maybe missing its melanin in its feathers. So the only reason that this bird is white is because it doesn't have the melanin to soak up all of those other wavelengths of light that are making it blue. So color is really complicated, it turns out. You're producing these patterns. We might think of it's just green, but green is actually made up of feathers existing in a certain place in a body that are a certain shape, that have a certain structure. And so, all of these are working together to produce these amazing patterns that we see. Now, as I mentioned, for a long time, people thought about structural color. They thought about pigmentary color, and there was a lot of foundational research that was really important for thinking about how bird colors are made. But they largely ignored microstructure, and so the shapes of these barbs and these barbules. And increasingly, we're seeing that these are actually really important in tuning the colors of feathers, the colors that are coming off of feathers. A couple of years ago, I talked about what are called super black feathers. These are feathers that are absorbing more wavelengths of light than they could with pigment alone. Here's an example of a superb bird of paradise, which looks like a black hole because it's got these really fun feather microstructures. And so this is a typical black feather. You can see this is the barb. These are the barbules coming off of it. This is a super black feather where the barbules are actually angled up and they have all these extra little projections. And so all of that is adding up to just absorbing light. So these birds are modifying the microstructure of their feathers and changing the color. Now, I talked about this a couple of years ago. That paper was being presented by a friend of mine when I was a graduate student at Harvard, Cody McCoy, and that led me to a lot of curiosity about these Ramphichelys tanagers that I had studied from my master's. I had measured them, and I had found that a lot of their readings were not what I would expect based on the colors that I was seeing. So, for example, this maroon silverbeak tanager was reading essentially as black with my spectrophotometer, something that had always puzzled me and led me to collaborating with Cody on a study of both the carotenoid pigmentation, again, carotenoids produce those yellows, oranges, and reds, and the feather microstructures of males and females from the genus Ramphichelys. There are 10 species there, to understand why are these colors different in these different birds. We found that actually carotenoids were highly correlated between males and females. I can look at these birds. These are the females. They look very different than the males, much less vibrant. And so there's something going on there. It's not the carotenoids that are different, but we found that actually the microstructures were very different. So these are the different shapes. This is cross sections. Here's that typical feather where here's the ramus of the feather barb. These are the barbules. They just lay flat. But in these really vibrant red feathers, the barb is actually really fat. It's expanded. The barbules are almost completely absent. And we showed that actually is making more red light go through that pigment and then hit that pigment and come back out. So that red is actually much more saturated because of the shape of the feather, not the pigments that are in the feather. We found that super black structure was in this really maroony, very interesting silverbeak tanager. So they angled their barbules, allowing them to absorb more light than just having those carotenoid pigments alone. Now, when we quantified across these species, these triangles are the males, these circles are the females. You can see the circles all clustered together here, where there's a lot of variation in the males. And so we concluded that a lot of these differences between these male and female tanager colors weren't the pigments that we're putting into them, but they're actually the shapes of the feather microstructures, which is very exciting. And that led me to studying this group of birds. I decided to collaborate with Rosalind Price Waldman, who was really the leader of a lot of this work, and Cassie Stoddard at Princeton. And we decided to start a project where we would sample many different regions from these birds, of course, not encompassing every single color that's on these birds because they are amazingly colorful. But we measured about 200 feathers from specimens from the Natural History Museum of Los Angeles County, my museum, the Academy of Natural Sciences in Philadelphia, and some in the Princeton collection. Now, what did we do? For each of those patches, we took a reflectance spectrum so we could get a really accurate measurement of color. We also sampled individual feathers and took pictures of them so we could get an idea of what the feather macrostructure was, how long were the feathers, how long are the barbs, how many feather barbs are there. We then looked at these feathers in the microscope so we could get a sense of, the shapes of the feather barbs and the feather barbules, so we could understand how much microstructure variation was there in this clade of tanagers. Now, I am not going to tell you the results of that research today. We're still kind of in the process of publishing that, so TBD to the future. But I'm going to talk about a story about these feathers that we noticed while we were doing these analyses that led us down a whole nother direction, which had a really, really cool finding. And that is this black spot. So in this crown feather of this paradise tanager, we were surprised to see that there was a really dark color underneath the bright green. And so as you might recall, feathers are composed of a downy part, that's kind of the gray fluffy part, and the colorful tips, and that's all anyone ever talks about when they talk about it. The more we looked at different feathers in these tangar tanagers, the more we realized that almost all of them had an intermediate region between the gray downy part and the colorful tips. And in structurally colored feathers, that region was black. You can see in this blue feather, here's that black region right before the downy region. And in carotenoid colored feathers, that intermediate region was white. So here's that white region, here's the downy region, and here are those colorful tips. This was surprising. Nobody has ever described that in bird feathers before. And so we decided we should study that. We should study what we're calling the achromatic region, which is either white or black, depending on the mechanism, the primary mechanism associated with the colors being produced in their tips. We thought that there might be some sort of visual application here. When you think about how feathers are laid on top of each other, this is a really, really, really close-up picture of a paradise tanager head. Feathers are essentially tiled. Think about like roof shingles or something like that, so that those colorful tips are actually laid on top of that achromatic layer. And so maybe they are modifying the color that's laying on top of them. TBD. So if, for example, this is a red-necked tanager, if we were to look at the achromatic layer that's between the downy layer and the colorful tips, we would see something like this, where underneath all of the structurally colored patches, we have black, and underneath all of the carotenoid patches, we have white. So we decided we should ask this question, which is, does the achromatic layer change the feather color? We decided to sample 12 focal patches from this group, 6 carotenoid-based colors and 6 structural colors from both male and female Tangara tanagers. Here are the amazing, beautiful tanagers that we sampled. They're from a variety of different regions of the bird, some from the crown, some from the rump, some from the belly. You notice that we did 2 from each region for each coloration mechanism. And we threw a whole bunch of really fancy equipment at them, which was very fun. There's nothing that I like better than looking at feathers under fancy microscopes or with really fancy spectrophotometers or really fancy cameras. So we use spectrophotometry, which I've already talked about. This is what the spectrophotometer looks like. Here, this is the probe that gets into the bird. That reading goes back to the spectrophotometer and into my computer. We use what's called multispectral photography. microspectrophotometry. I'll talk about this in a second. Microspectrophotometry, think a spectrophotometer, but on much, much smaller level, so we can measure the colors coming off of individual feather barbs instead of big, giant regions of feathers. And a hyperspectral camera. So this is, think about if a camera and a spectrophotometer had a baby, it would be a hyperspectral imaging center or a hyperspectral camera. So we're getting reflectance spectra at each pixel of a camera. Okay, first we're going to talk about multispectral photography. So this is photography. So your camera sensor is actually taking in different sorts of channels of light, typically red, green, blue. Again, corresponds to our human visual system, so it makes sense. We care about what we can see when we're thinking about a camera. But we can modify cameras to accept ultraviolet colors. So this is an example of a paradise tanager from our collection. And we're seeing reflectance in these different wavelengths in addition in the ultraviolet. So what we did was we took pictures of 3 feathers from each focal patch on both white backgrounds and black backgrounds. So this was simply to ask, does putting a feather or putting these tips on a different color background change the color that's perceived? A lot of this was done by Jerome Ali, who was a member of Cassie Stodder's lab, who was an expert in multispectral photography. And then from each photo, we measured hue, chroma, and lightness. So if you're not a color person, hue is basically what we think of when we define what color it is. So think blue, blue-green, green, yellow, yellow-red. What we're describing there is the hue. Chroma or saturation is how rich the color is. So it can range from being not very saturated at all. So this might be, you know, a very washed out blue to being a really rich blue that is highly saturated. And then lightness is just what you think it's just how light a color is. Also known as brightness. I'll use those 2 terms a little bit interchangeably, but when we're talking from a camera perspective, lightness is technically what we're measuring. So this is what those photos look like. Here's an example of a carotenoid feather and a structurally colored feather on a white background and a black background. You can tell that there's probably going to be some impacts of the color of the background on those feathers. Here's an example of, so I said for each patch, we measured 3 feathers per patch. So here's the rump feathers of a paradise tanager. Now, here are the results from each species. There are a lot of graphs on here, and I don't expect you to look in detail, but the key thing I want you to take away is that they are very consistent. Each row is a species, each column is one of those measures. So hue, saturation, lightness of both the carotenoid and the structurally colored feathers. So consistent amongst different patches. It doesn't really matter which one we're talking about, the results are the same. So let's pick 2 examples to go through what are the patterns that we're seeing. Well, let's focus first on carotenoid pigments. For both carotenoid pigments and structural colors, hue remains the same. So the actual hue, or how we're defining the color, doesn't change if it's on a white or a black background. But in carotenoid-based feathers, lightness, or how bright a color is, does change. So it's much darker on a dark background and much lighter on a light background. Now, this is because you might think of feathers as being opaque, but actually feathers, there's a lot of light that transmits through a feather. And so it's not just the feather itself that's producing color, but there's also a major effect of that background. We're showing that here. Saturation doesn't change. So really, for carotenoids, it just seems to be affecting lightness. Now, for structural colors, lightness is also affected. So on a black background, the colors are darker. On a white background, the colors are whiter. But interestingly, saturation also changes. So if you remember, when we're producing structural colors, there's a layer of melanin that's important for absorbing all of the extra wavelengths of light that are basically making a color appear kind of muddled. So it appears that that black background is actually contributing to that. They're making that structural color appear even richer by helping to absorb those extra wavelengths of light. Now, you might be thinking, okay, Allison, this looks promising, but what if there's something to do with how those barbs are? What if it's like a combination of all of the feather barbs instead of just an effect of single feather barbs? Well, that's where the microspectrophotometer comes in. This is an example of what we do. This is one feather barb of a blue structurally colored feather, and this black dot is the precision with which we can take a reflectance spectrum. So here we're just going to show it's not just photography, but actually with a spectrophotometer, does it change that? Now let's just take a step back and let's remember what we might expect a reflectance spectrum to look like. So remember, as we said before, we've got reflectance, or how much light is coming off of it, on the y-axis, and we have wavelength of light on the x-axis. This is just kind of an example of what I might expect a carotenoid pigment reflected spectrum to look like. Well, what I might expect something that gets brighter to look like, I would expect there to be I would expect a higher level. So we would expect more light coming off of it. If something is darker, we might expect less light coming off of it. So I might expect a pattern that looks something like this if background is important. This is an example of what a typical structural color reflectance spectrum looks like. And similarly to the carotenoids, what do I expect to happen? Well, I might expect if something is lighter, for there, again, it might overall be taller or not. If it's darker, I might expect it overall to be taller or not. But the key here is actually how I would expect this in order to read saturation. We actually see this peak is more distinct compared to all the other wavelengths of light. So this peak is taller when compared to how much of light is coming over here. So in this top one, which is kind of our most washed out blue curve, this peak is not much different than this rest of the reflectance spectrum over here. So that's going to look much less saturated. So that's just a little example. Now, what do we see in our real data? Well, with the carotenoid pigments, we see just what we might expect for brightness to increase. So we see much more reflectance in the yellow part of the spectrum when it's on a white background compared to when it's on a dark background. And with the blue, again, we see it actually most of the difference is in these part of the reflectance spectrum that are not blue. So it's either light that's being kind of captured by that black background versus light that's it's still being scattered back through that structural color. Remember, these feather barbs still have melanin, so they're still doing their job of absorbing all of the non-blue wavelengths of light. It's just some get through, some don't get captured by that melanin for whatever reason. So the black background appears to be helping with that. Just like with photography, the results were incredibly consistent across all of the different species. So I'm not going to show you all of those results again. Just know that these two examples basically represent what we saw across all of the different birds that we measured. Now, if I haven't convinced you yet, maybe you say, Allison, you were putting these birds on artificial white and black backgrounds. Well, we can look at what these look like. So this calibrite backgrounds are what we used, and we compared them to the black of an achromatic region from Tangara chilensis, or we compared them to the white of the achromatic region of Tangara chilensis. You can see the curves look about the same, but there are some differences. So we thought, okay, let's use an achromatic background that's actually from the bird itself, but actually do some controlling. So what we did is we took a specimen and on small windows, we trimmed off the colorful regions. So we trimmed off the colorful region on the rump, and we trimmed off the colorful region on the crown, so we could have a real white and black background. And then we could put feathers from this species on top of that and measure how does having that color actually or how does having that actual true white and black background impact the color that's coming off of them. Just for if you want to see what this is what it looks like. For those of you who might be concerned about the specimen, this was essentially a no data specimen, so we don't know exactly where or when it's from. So it's not super useful for science, but it's very useful for this. So we decided to allow this particular use of this specimen. This is the hyperspectral camera, which is run by Ben Hogan in Cassie Stoddard's lab. He's a hyperspectral camera expert. It's actually a very temperamental piece of equipment and quite difficult to use. So we were glad to have Ben on board to help us with this particular project. So here's an example. We took a feather from the belly, and these are the feather barbs on top of that little black window. And so what did we see? Well, we saw essentially that the results held. So everything that we saw when we used artificial backgrounds were still true. So we did one more thing, which was we created a mathematical model. This is Jerome Ali and Roslyn and me. We have our fun lab goggles on because we'd also been doing some imaging. But on this whiteboard, we wrote out on this trip to Princeton that I did the first kind of attempt at this model, which was based off of a model of skin that had been published of lizards, where we had compared, we modeled basically how light would go through different types of colors and be refracted back off of that. I'm not going to go into all the math of that. If you're interested in that, happy to send you that later. But with our model, when we simulated what would happen with light, we once again recapitulated exactly what we found with our actual birds. So with all that, we put it all together. We use very fancy methods of very fancy imaging, kind of all the different types of backgrounds. We looked at whole feathers, we looked at barbs of feathers, we looked at the mathematical model, and all of that said the same thing, which is that having a white background with carotenoid pigments makes your color much brighter, and having a black background underneath your structurally colored tips has much changes the brightness is lower, but it also makes your color much more saturated. So hopefully you're convinced that when we ask this color, does the achromatic layer change feather color? We can say, why, yes, it did. It changes it in those ways that I just described. But this opened up a whole slew of other questions with us, which is, you might say, but Allison, is this just a developmental constraint associated with coloration mechanism? So maybe when you have carotenoids in your feather, you always are going to have that white layer because you just need that for putting down carotenoids. Or maybe if you have a black layer, you always need that for when you're putting down carotenoids. You might wonder, is there variation among species, or is there variation among individuals within a species? Does this phenomenon exist outside of tanagers? Are we talking about something that only Tangara tanagers do? Well, to answer that first question, we looked at feather edges, the edges of different patches. So these are feathers that have blue tips, but might be overlaying the orange of the carotenoids, or feathers that have black tips but are overlaying the red. If in fact our pattern holds true, we would expect this first layer of red feathers to have black. So those blue tips could be on top of black instead of on top of white. We would expect this to be the pattern in most of the red. We would expect this to be the pattern in those tips. Down here, we would expect those black tips to be over black to make them even blacker instead of over white. And in fact, we found this exact pattern. So these are what we saw in the tanagers themselves. So in the most of the patch, the achromatic layer was white. In the part of the patch that has blue laying on top of it, the achromatic layer is black. So, and same thing down here when we're looking at these rumps. So, is it just a developmental constraint? I think we can pretty confidently say no, that achromatic regions from patch edge feathers match the color of the overlying feathers, not their tips. So that's pretty cool. Now, is there variation among species or among individuals within a species? To answer this question, we looked at the proportions of these different feather regions across both males and females, and then amongst all the species. So now we're going to look at the proportion of the feather that was the colorful part, the proportion of feather that was the achromatic region, the proportion of the feather that was the downy region. For now, I'm going to focus on these top two because this is just, you just subtract the other two from the whole length of the feather, so they show the same thing. Each of these is one of the feathers that we measured. So each column represents a different species. The triangles are the males, the circles are the females. The key things that I want you to take away from here are that in some of these species, there are major differences between males and females. So here's the females, here's the males. And in fact, so there are differences in the sizes of these achromatic regions. And in a few of these species, in particular, for example, Tangara arthus, Tangara celadon, actually the females have black where the males have white. So that's pretty cool. That shows that there are differences between sexes in addition to among species. When we look at these, one thing, if you're very familiar with Tangara tanagers, you know that the males and females are fairly similar, but in many cases, the females are simply less vibrant versions of the males. Here's an example of pictures of some of those that we found these differences in achromatic layer. And so when we measure these different regions with our reflectance spectrophotometer, we can measure. This is the male. Generally, the male is brighter and often more saturated than the female of those same different regions. Okay, what's really cool is where we see the greatest sexual dichromatism in this group, we also see the greatest differences in these achromatic regions. And in fact, when we take those feathers and we put them on different backgrounds, if we put them all on the same background, the differences between males and females essentially go away. So only when we put the feathers back on their appropriate background for the colors of those intermediate regions they have, do we recapitulate the differences that we observe when looking at these whole specimens. So it appears that actually these birds aren't changing the pigments that they're putting in their feathers. They're simply changing the color of that underlying achromatic region to make the differences between males and females. So is there variation? Yeah, there's actually achromatic color differences that cause sexual dichromatism in some species. Now, that's really important from a behavioral ecology perspective. There's a lot of focus on how colors can actually be representative of bird health, they're going to help birds to choose their mates. And so now that we know there's all of these different ways to tune their color, if you remember a long time ago when I talked about those Ramphichelys results, those birds were tuning their color based on the microstructure of their feathers. Well, maybe these birds are able to tune their colors in a lot of ways that we're not accounting for when we're thinking about how color is made. So this is very cool. Opens up a whole lot of questions for future research. Does this phenomenon exist outside of tanagers? And we surveyed a number of different birds from different families that have these similar colors. And we found that, yes, there are achromatic regions in very brightly colored birds like cotingas, birds like manakins, and there are no achromatic regions in a lot of, closely related birds. So it appears that these birds appear to be evolving this across songbirds to modify their color. Interestingly, there's also variation within birds. These are feathers from house finches and from northern cardinals. There's no achromatic region in the crown, but there are achromatic regions in the rump. Now, interestingly, cardinals, they have a crest, so you wouldn't have the feathers laying on top of each other. So is that why they don't have the achromatic region? I don't know, but it's very interesting that we see variation even within a species. And actually, we see some species using it in the ways you wouldn't predict to make their color more vibrant. So maybe they're actually using these achromatic regions to tune their color in other ways. So for example, our friend the silverbeak tanager, which has that super black color, it's also got a black achromatic region. So that's probably contributing to this really interesting maroon color that they have. A couple of these really beautiful fairy wrens, the superb fairy wren, the variegated fairy wren, these have structurally colors and they have white achromatic regions. So maybe they're trying to show their color off in a different way. I don't know. But what's clear is that does this phenomenon exist outside of tanagers? It does. It's broadly distributed across songbirds, across passerines, but many exceptions to the rule exist. So I think there's a lot more to be studied here. I think we should consider, the entirety of feathers. something that I'm excited about thinking about in the future is, is there any other function of the achromatic layer? We talked about how it's modifying color, but does that change anything else that a feather is do? If you know me, you know that I'm really excited about these functional trade-offs when you change one aspect of a feather trait. What else are you changing? The last time I talked to you, I talked about how the proportion of the downy part of the feather changes in different elevations in the Himalayas. And also we looked at the color of the downy part probably plays a role in thermoregulation too. So we know that the color of the down changes with the overall body size of the bird. So I don't know. There's a lot to be done here. I don't have any answers for you other than what I just presented to you, but I think this is a very cool phenomenon. And, as we're studying the evolution of these color patterns, we really should be studying all of the things that are going to make up this entire color pattern. Now, I just want to end with kind of bringing us back to the art world. So I talked about super black color. If you're familiar with Vanta Black paint, this is a special paint that uses carbon nanotubules. That's essentially using the same concept as super black birds use. I know, Max. But we also see the achromatic layer being used in painting. Grisaille is a method is used for many, many years, used by the masters, where they would actually do an underpainting in whites and blacks. Bright colors were extremely expensive, and so it was actually much more affordable for them to do an underpainting in whites and blacks and then glaze over it to produce the vibrant colors that you see in a lot of the famous paintings today. Glassblowers, Dale Chihuly developed what's called a cloud layer technique to keep colors separated. So he would blow a white layer of glass in between brightly colored glass of so to keep those colors separated. And finally, if you're a makeup person, you know that adding a white base to the foundation before you put your foundation down can help produce what they call flawless skin. There's a whole TikTok I went down a long TikTok rabbit hole on the white foundation base layer. So highly recommend looking into that. And finally, if you ever go to a jewelry store, they will show you gemstones on a white background. This is an example of an opal, and you can see how, again, all of that color that's refracted off essentially gets washed out when it's on a white background, or this moonstone, when it's on a white background, you don't see that beautiful blue. So the color of the background matters. And, this is something that we are learning from looking at museum specimens. So if we're just looking at individual feathers alone, that actually is not going to be representative of the entirety of a bird's color. So I'm always excited to explore the collection and see what new things we might learn from the collection. Of course, many acknowledgements here. A lot of this work couldn't be done with the advice or input from many different people. Of course, the collections managers who allowed us access to their collections, and advice using all the fancy microscopes that I talked to you about today, and of course, our funding. So I'm going to leave you with these main conclusions and hopefully get you thinking about after this. You're going to think about what's under the layers of a bird that I can see. And I am happy to take any questions that you might have. Thank you so much for listening.
Fantastic, Allison. Thank you very much. It was wonderful. If you
Yes,
have any
great.
questions, please pop them into the Q&A section. It's down at the bottom of your screen. That helps us keep track of everything.
No, and we already have one. John asks, how uniform are the achromatic layers across different regions, and does that change as the bird moves? For instance, you might expect the rump to remain more consistent than the wings.
Absolutely. That's a great question, John. And I think, as we're talking about something like a cardinal or something that has used crests, I would predict that we wouldn't see the same achromatic regions in anything that's going to be changing shape, or feathers that are going to be lifting up in the way a crest does. I wouldn't actually expect to see achromatic regions in wing feathers, although maybe epaulets, although it would actually be very interesting to look at epaulets, and sometimes they can be puffed up and sometimes not. But this is the first time anyone's actually described this. So TBD. I think we need to learn more. So I see there's another question. Have there been any studies on the reflectance spectrums of the surfaces of other animals like reptilian scales, fish scales, amphibian skin, or insects? And the answer is yes. There have been studies. There's a lot of people that are studying, reflectance spectra of these different animals. There's different sorts of challenges that are associated with other animals that are not birds. So for example, if you go to our fish or herpetology collection in the museum, a lot of those specimens look completely different colors than they were when they were alive because you need to be living in order to have those pigments. And when you put them in alcohol or you put them in formaldehyde and then alcohol, that's changing the composition of those pigments completely. So you're really not seeing what's alive. And so, measuring fishes underwater, special technical challenges that are associated with that. Measuring really tiny insects, special challenges that are associated with that. I have a collaborator from CSU Dominguez Hills, Terry McGlynn, who's my main collaborator on infrared reflectance. And we got a very, very small, tiny probe for measuring ants, which is Terry's specialty. And still, it turns out that I think photography is better for measuring the ants. So yeah, definitely people use them, but different ways that they're using them.
Interesting.
Yes.
Have you ever thought about taking this beyond to non-passerines?
Absolutely. That's,
Yeah.
I think, would be a very cool study. Just when I was doing broad surveying of the collection to gather some photos for that figure, one thing I kind of learned was non-passerines don't seem to have the achromatic region. And so this was just me looking at some of the looking at some bee eaters, looking at some kingfishers, looking at some toucans, other brightly colored birds. They seem to have something else going on completely. So again, just exploration, really fun, but lots to be done.
Interesting.
Yeah, so Charles Hood asks, do you think the leucistic Steller's jay knew it was a different color? And either way, how did the other jays react to the pale one? I don't know the answer to that because that was before I was really an ornithologist. There are a lot of people there are a lot of other leucistic birds out there, and we know about more and more of them because of iNaturalist, because of eBird, because of people like you all out taking pictures of these very interesting birds. There was a long study of the yellow cardinal. It had something wrong with the enzymatic pathways that make the carotenoids red instead of yellow. And I can't remember now if it found a mate or not, but color is really important for bird communication. So
Yeah.
I can imagine that they would have some problems. People that other birds might not recognize them as that species. One of my favorite studies actually on melanin pigments was on this frigate bird that washed up on the East Coast that was a leucistic frigate bird. So it was missing all the melanin in its feathers. So in addition to providing these optical properties, melanin is also super important for strengthening feathers. And so this frigate bird, which is exposed to salt water and all sorts of really rough conditions, its feathers were destroyed. It had essentially in the tail, there was only the rachis left. Almost all the barbs were completely gone. So I think this bird, its feathers had just gotten so worn down it couldn't fly anymore and eventually washed up. It turns out that specimen's in our collection. I discovered this like a year ago and it was very exciting. But I think, these when I'm talking about these feather color trade-offs, that's one of the things is, when you change the color, it's not only the color, but it's other aspects of the feathers too. So good question.
Right.
So another question, have anyone been able to find the patches on any other bird groups besides passerines? As I mentioned, TBD. So hopefully we'll be able to work on that in the future. Do the species I presented here undergo distinct molts like many gulls and ducks do? So there typically is an immature or a juvenile plumage in these birds. Oftentimes that plumage looks a lot like the female plumage. They don't go through, years of different molts like we see in many gulls and ducks, but yeah, all of this work was just one male and one female. So there's a lot to be done on how much variation exists across life stages, exists across individuals. And I think, members of my lab are going to be doing a lot of that work in the coming years. So just had a grad student, we just have a grad student who's working on female microstructure and is going to do a lot of this complementary work. So really trying to understand, why does dichromatism happen? I showed you in a few different individuals, but there's a lot of variation there that's not accounted for by the achromatic variation alone. I have a postdoc, Jenna McCullough, who just started, who's going to be studying structural white color, which is very exciting. And so that's, birds that are producing white in unusual ways. So, yeah, there's a lot to do and a lot that we're going to do, and I'm very excited about all of it. So, we'll have more people to come present more LA Bird webinars, and you'll have to just keep having me back if you want in coming years to.
Of course.
Well,
Can I ask one follow-up question?
I think you said TBD about 6 times tonight.
I know. It's just exciting.
Yeah, so that's 6 more webinars.
I can't wait to have answers to these.
Allison, can I ask you a follow-up question to that?
Sure.
I'm wondering that everything you've presented tonight argues that there might be different mechanisms for sexual selection acting on the colorful portions and the achromatic regions, and clearly there are selection pressures happening, as you mentioned, on the achromatic regions or the inner parts of the feathers. If you had to make a guess on sort of what the genetics and how selection acts, is there any sense, and I'm sorry if this knowledge is out there and I don't know, but are there genes linked for the different areas of the feathers? Is it possible they're under completely different selection pressure?
Absolutely. We actually know very little about the genetic basis of barb and barbule morphology. So we know, there's a lot more work that's being done on the genes that are turning on and off carotenoid coloration, that are turning on and off melanin coloration. But we know very little on the genes of structural coloration, very, very, even less on the genes of like barb and barbule variation. So, I think one thing to be done is actually really characterizing these achromatic regions. Are they just adding and removing melanin pigments, or are they actually changing the shapes of the feathers and as well, the microstructures, to be more or less reflective? Clearly they're doing something, and I don't know exactly what that something is, but I would speculate that there's definitely selection working on this. We wouldn't see these differences between males and females if there wasn't. And yeah, I'm excited to learn, I want to point our infrared spectrophotometer at these and see if there are heat implications, for their feathers' ability to absorb or reflect heat when you're changing these colors as well. So yeah. I don't know. So much to do. So little time.
A lot to explore. That's wonderful. Well, I think we've run out of questions for the moment. Thank you very much, Allison. It was a wonderful, wonderful webinar.
My pleasure. Hopefully it wasn't too many figures and graphs for you.
No, it was great.
My first time presenting this, so
it was great, and we're looking forward to the 6 new TBD webinars that are coming up.
Sounds great.
Absolutely. And also, one of the wonderful things about recording our webinars is it gives people a chance to go through it bit by bit or over. And it's a great learning experience. And I think we had another question pop up.
Question.
Oh, thanks, John. He was just agreeing. It was a wonderful webinar. Yes. Thank you all. Thank you very much. We'll join you for, please, you won't join. Please join us for our next webinar on October 14th. And until then, good night, everyone. Thank you all very much.
Thank you.
Good night. Thank you, Allison.
Bye.
Bye-bye now. Thanks again.